Okay, so I guess I could get started a little bit here as people wander in. Just to let you know, some of the summer students have started this week as opposed to last week. So if you missed the first two lectures, they actually are online already, I think. I'm pretty sure they are. And you can find them on our fMRIF website. I think so. Most of you should have gotten. So just a reminder. So either I'm sending it out to. There's a listserv called fMRI Discuss or a listserv called fMRI or 3T Announce that I'm sending things to. But I'd like to send them only to the fMRI_course listserv. So you could go onto our fMRIF site and log on to that listserv. And also that's where you'll find not only the latest lectures, the last two ones that I gave last week, but from 2015 and 2014 lectures. And not only do you get the movies and the lectures in real, sort of the progression of the lecture, but you also can get the PowerPoints just for reference as well. So we have both those downloads. And yeah, that's thanks to Rock Mercado, who is our IT person for our core. So, okay, in the first two lectures, one was on sort of the history of neuroimaging, and the other one was on fMRI limits and contrast mechanisms and things like that. And this one actually, so those two things are sort of, I know very well, and this sort of I know very well from my own perspective, but one thing that's a little bit different between myself and maybe any other person talking about processing methods is that I'm not really I'm not a statistician. I don't even like statistics that much, but obviously with fMRI you have to live with that, and it's becoming much more important. My feeling about statistics, though, is generally that if something is extremely dependent on exactly the statistical test that you use, the result probably isn't strong enough to really believe. So I mean, that's sort of an extreme view. I don't really have that view, but I kind of like to say that for people who say, well, if I do this sort of test, then we get a result. But if you do this test, we don't get a significant result. I'm thinking, well, let's just sort of redo the experiment then until we get a result that we can actually rely on. So my view on paradigm design and processing methods is more from sort of a creative standpoint of, okay, what things can you do as far as inputting brain activity and pulling out information. So that's sort of like the general gist of what I'm trying to talk about. Okay, so for those of you who were here a couple of days or last week, you saw this sort of diagram. This is my four major pillars of fMRI, that it's driven by the technology, the methodology, the interpretation or better understanding of the signal, and also the applications. So the main thing I'll be talking about today is the methodology, and is in terms of paradigm design and processing methods. So there's three main parts of my talk. I'll be spending a lot of it on neural activation and input strategies. Then I'll have a very brief introduction to resting state fMRI. I would say about a third of the lectures in this course will be related in some way to resting state because it's become so important. An aspect of fMRI that's sort of taking off. And then I'll talk a little bit about fMRI decoding in the sense that it's a very different way of looking at the data, and it has its own questions in itself, and its own issues, and it's still growing rapidly. And it's also really kind of a cool thing to look at. Okay, all right, oops. All right, so this is a summary slide of at least my worldview of the various types of neural activation input strategies that you can do with fMRI. And this is somewhat it's not really all-inclusive, but it follows a sequence of, the early studies have included like block design. The basic idea with block design, you would have, for instance, 20 seconds off, 20 seconds on, 20 seconds off, and so on. And the reason why you would modulate at that high of a frequency, it was considered high relative to PET scanning, where you would have maybe 5 minutes off or 5 minutes on, is because the baseline with fMRI does drift around a lot. And so if you actually have something changing much more rapidly than the baseline of the signal, you can separate those two frequencies very easily. And that's the basic intuitive strategy of that. Second thing I'll talk about is frequency encoding. This one is really esoteric. It's really just something to sort of expand your mind, but it's not really something that is done at all. It's just wanted to just demonstrate that how it can be done. It uses the basic encoding, basic signal processing approach of multiplexing, putting in different frequencies into a time series and being able to pull them out and then separate them cleanly. The third is phase encoding, and that is less esoteric. It's used primarily in the context of something that if you continuously vary some aspect of the stimulus, the spatial location or the spatial extent would change and also in a continuous fashion. So it's very powerful because you're constantly driving the system and you can actually pull out the relative information based on the relative phase of what part of the signal occurs over time. I'll explain it more as I go into it. And then event-related came along, and that was so that sort of revolutionized fMRI a little bit, being able to, instead of giving a block design and forcing a subject to do something for like 20 seconds or so, you can actually give a very brief stimulus. At first, we actually thought we only had to give a very brief stimulus every like 15 seconds, but turns out, as you design the paradigms, as we would use very basic signal processing strategies, you can actually go much more rapidly with fMRI with event-related. So beyond that, there's no iconic figure for fMRI adaptation, but it's another strategy that's used a lot or somewhat by either psychophysicists or cognitive neuroscientists. And the basic idea is trying to pull out areas even within a voxel, an imaging, sort of three-dimensional imaging area that's very small, where some neurons fire to a certain stimulus, and if you give that same type of stimulus again, they'll have a certain refractory period where they won't fire as rapidly, and the fMRI response will be dampened. So the idea is that you give a response, and you try to basically see how similar the stimuli are as the signal either goes down as it adapts or not. So I'll talk a little bit more about that. And orthogonal block design. So basically, any time series that's orthogonal to another time series, the correlation is by definition 0. So that means that, for instance, these two time series right here are approximately orthogonal. And if you calculate the correlation coefficient between the two, they'd be 0. So in other words, if you had two paradigms that were exactly shifted by 90 degrees, like these are, you would find that if you had a response that was ideally time locked to the paradigms, you could put in a reference function for one, and then put in a reference function for the other, and have absolutely no overlap. There would be no spillover of the effects because they're orthogonal. And so you might want to design paradigms in which you have several different types of stimulus timings that are all orthogonal. So you can just put in these different time series and then pull it out. And then the other thing, though, is that you could actually, there's a function on AFNI. It's a standard statistical package where you can orthogonalize any function. Even if it's not orthogonal, you can make it orthogonal. You take a hit in statistical power, but you can do that still. And that's actually what a lot of people do to sort of clean up their data a little bit better, a little bit more. And then there's free behavior design, which is something that I really think is something that will still grow a lot. And that's basically having a subject in the scanner freely behaving, either watching a movie, doing some sort of real-time feedback where they're trying to change their brain states. The key to free behavior design, though, is to really keep close track of what they're doing and use whatever measure of what they're doing as a reference function. All right. Okay, let's just begin quickly with block design. So block design, this is way back 23 years ago. I was lucky enough to be able to write this paper on processing strategies for time course data and functional MRI of the human brain. And I just compared various block design paradigms for the most part. And the idea was, up until then, fMRI existed for about a year. Up until then, people were either doing subtraction. That's where you basically, within a time series, you take either one image or an average of images and try to subtract it. This corresponded to this. If you average a bunch, that corresponded to that. And then people tried to put in either a boxcar, and they figured, oh, that works pretty well, and that's this. I'm sorry, that's, actually I have the arrows right here. Yeah, so this corresponds to, this is basically this is the raw data from the left and right motor cortex. I'm tapping right fingers and then left fingers and right fingers and so on. And you can see they're exactly out of phase. One shows a negative correlation and a positive correlation with one of these data sets. And then you put in reference functions. Sometimes you can just have the boxcar because it doesn't really matter that you don't really have to smooth the function that much. Or you could put in a Gaussian smooth function or one that's convolved with what's known as the hemodynamic response function right here. So you can actually put in any one of these. You can create these functions in NAFNI as well. I think there's a function called waiver that creates any of these functions you want by putting in the input and finding the smooth functions. But then you can do very basic Fourier analysis. So if you take the Fourier transform over this time series, you get something like this that has the predominant frequency. This happens to be the on-off frequency of 0.031 hertz. And you could decide to look at the power under this curve, under this area, or just use this as the reference function. And the reason for that is because it's a boxcar right here, because there's all these other this signal is not a sine wave, so it has a bunch of different frequencies in it around these edges. And so you have different harmonics as well, and you want to include all those. And so that is basically that right there. So, I was basically playing with the various input functions and finding that, for the most part, it doesn't make a huge amount of difference what you do. It helps maybe to be careful of the delay. But the delay does vary over space. So there's a lot of different hemodynamic response functions that might cause a delay over space. And so fNI also has that function where you can put in a function, a reference function, and jitter the delay and actually find whatever correlation is highest for a given delay. It increases the power a little bit. Okay. All right. And so what's interesting, though, is that here was very simple. I was tapping my fingers. And this actually was my brain. I keep on saying me because when I was a graduate student, I put myself in the scanner, analyzed my data, and then published the paper. But you can use a simple model like this on-off function, and that's just left and right motor cortex. I didn't know to put in any model because I wasn't expecting the brain to be behaving in any other timing. But in fact, it probably does. No matter what block design you put in, the brain is probably doing more complicated things at different timings for different areas. So here's a really nice example. And I showed this actually the other day, but I want to re-emphasize it, by Camille Uludag. And here, very basic paradigm. You're putting a single hemifield checkerboard stimulation. And that's known to stimulate the opposite visual cortex. And if you do that, with a standard response model, as what's expected to turn on when you're shining this checkerboard, you get something like this, which is what you expect. The other hemisphere is activated. If you put in something that's a little bit outside the model that most people don't quite expect but still happens, is you put a transient signal change at the very beginning, a transient at the very end, and just use that as a reference function, you get something like this, which is very different than this map, but it's still meaningful brain activation. It's bilateral, more peripheral, or maybe even MT, or who knows what exactly, visual cortex that's showing activation. It's very clear bilateral and insular activation. So there's a whole different hypothesis that's generated by these two transients. So it's very important when you're designing how you analyze your data to not just put in a very simple function, to sort of maybe consider other functions to put in. All right, and this I also showed you that if you really average the heck out of the data, this is average over 9 hours, the subject came back multiple times, you pretty much get most of the brain doing something. and we're still in the stages of trying to interpret what this means. So this subject stared at a stimulus, tapped their fingers when they tapped a button when they saw either a number or a letter within the visual stimulus. And you get what you'd expect. Visual cortex shows this sort of expected response. Motor cortex shows something like this, but then you get all kinds of things: negative responses, negative and positive responses, and these are all probably real. This was averaged for 9 hours, and if you look at the pattern, it does correspond to functional units in the brain. You find areas of the default mode network that show a clear decrease in signal during the task, which is known to happen. When your brain is doing something, there's less mind wandering. At least that's a hand-waving argument that the default mode network is related. It always generally is turned off during a cognitive task. And this is exactly what we see here. So if you want to probe that, you need every one of these reference functions. And it's hard to generate these because there's no way that you can really have a clear hypothesis for all of them. Anyway, that's where the state of the field is right now. So frequency encoding is, like I mentioned, a very esoteric thing. We just want to do it because we wanted to see if it worked. Once again, this was 23 years ago, and we were just trying all kinds of things. So we had a subject actually first for cued to tap their right fingers and then tap their left fingers at various on-off frequencies. So they were verbally cued: start tapping right, stop tapping right, start tapping left, stop tapping left, and they were doing this in an interleave fashion. So you have two different frequencies. Take the Fourier transform of these two time series, you have two different peaks, and you can clearly see left and right motor cortex from the peaks right here. And that's just an example of, but you can imagine though, doing this sort of thing. I mean, people do this all the time with EEG. They do multiplex signal input with EEG. And so you can imagine even doing a paradigm where you can have different frequencies and on-off frequencies going in that's behaviorally relevant that you can actually pull out potentially. Okay, phase encoding. So phase encoding made its mark in the early days of fMRI when people were doing retinotopy. And the basic idea is that if you varied the phase of the location of the stimulus, you could rotate a stimulus and then map the activation as a function of what angle of rotation occurred, you can actually see the activation moving around with the rotation. This is a nice, just one of several examples. This happens to be Ted Dio, who I collaborated with, basically mapping out retinotopy from center to periphery. So basically having a ring that expands out and then repeats, expands out over a minute and then goes back in, expands out, and you're basically mapping. The color is coded based on what the angle, how peripheral the ring is. And you can see a very nice correspondence to this. The really interesting part of phase encoding is this map, and that is where we map out with the rotation where you can actually map out the visual borders. And the way that you do that is that with V1, turns out that the progression of the stimulus is in one direction along cortex, but in V2 it's a mirror image. So at some point they hit each other, and then they cycle through and they hit each other. And you want to find these where you can find the edges of V1, V2, and V3 because they're all organized in a mirror image fashion. So you can look at the progression of how the phase changes and then see how it starts over again and then map out V1, V2, and V3. It was a really elegant experiment. I think Marty Serino had a paper in Science on this. And Steve Engel had something as well. So then you can do auditory frequencies as well. So basic experiment was basically sweeping up or down, and they were able to make these really dramatic maps. And this is actually tonotopic mapping right here. So if you look at this, you can actually see very, very detailed maps of tonotopy right here as it sweeps along from 1 kilohertz up to 40 kilohertz. This happens to be in a rat brain, showing beautiful, beautiful results. You could never get this in this amount of time by just doing like a block design, like low frequency for 20 seconds, wait 20 seconds, higher frequency. That wastes a lot of time. So if you just keep on driving the stimulus, sweeping across the frequencies, that's a really effective, powerful way to do fMRI. And the same thing was done in humans. You can map out tonotopy and cortex, and also you can map out all the way down to the subcortical areas as well. And it turns out when doing this sort of frequency sweeping, we realized that the subcortical areas also have very clear tonotopy. And as I showed you the other day, another way of showing orientation columns within V1 is by, if you have a rotation of a grating that looks something like this. This is, unfortunately, I couldn't match exactly the speed, but it just basically shows that the area of activation within an orientation column follows this orientation grating. And so that's basically the orientation pinwheel organization of orientation columns, which was a beautiful experiment that worked really well. So event-related. So with event-related, came about in about 1996. Randy Buckner was one of the first people to actually demonstrate it, basically putting a region of interest in the left inferior frontal gyrus and actually doing a task which was word stem completion. So they would see a word like C-O-U, and they had to come up with couple or whatever. And so when they did that, their left inferior frontal gyrus became extremely active, and it was clearly shown as an event-related response. Okay, and then people actually were trying to probe this a little bit and found that things were kind of nonlinear. And this is an early example of showing if you have a linear system, this is what your expected response is from event-related relative to block design. So a block design is at 1. Event-related is right down here, so at about 0.3. So 30% of your contrast at best, which is not very good. It turns out that if you actually do this in real life, the actual contrast that you're able to get with the optimal ISI, this is using a constant interstimulus interval of about 14 seconds, peaks out at about 65 or 70% of a block design, which is way better than what you'd expect from a linear system. So the system's somewhat nonlinear, so the bold response favors brief stimuli in a way that we don't completely understand yet, but it's at least a good thing that we can actually see this. So, and here's an example of comparing maps, visual cortex and motor cortex, unthreshold, just grayscale, with block design, and then with the various timings of the interstimulus interval of 12 seconds, the stimulus duration of 2 seconds, and then so on. You can see qualitatively things drop off a little bit, but not that much, and then they start really dropping off at about a 6-second ISI when things become saturated. Okay, but then Anders Dale came along, and Randy Buckner, and there's another guy, Greg Burrock, who discovered that you can actually treat the visual system, or any system in fMRI, hemodynamic response, as a linear system for the most part. It behaves within a certain range like a linear system. In other words, if you give 2 stimuli that were only 2 seconds apart, or give 3 stimuli that are 2 seconds apart, you get this. So here's 1 trial, here's 2 trials where they add up, and here's 3 trials. And it turns out if you take this first trial response, shift it, and subtract it from the 2 trial, you'll get the complete hemodynamic response back. There's not much of a distortion of that. And that was a wonderful, wonderful thing, because that allowed you then to do all kinds of tricks, like give stimuli that are brief, but if you jitter the interstimulus interval, you have enough information then to pull out the entire hemodynamic response from many more trials than you would with block design. So, okay, so then people have written papers. This is Rasmus Byrne, who was a postdoc here at the time, did one of them, and of trying to understand, okay, what is the relative detectability versus the average ISI for this jittered responses. And so that's why there's this cloud of data, because he was doing simulations where he did multiple iterations. You can't actually, each iteration ends up with a different sort of optimal stimulus. But at least you get the idea from the cloud that, okay, so these solid lines are constant stimulation, or constant ISI, the old-fashioned way of doing event-related. And these are jittered interstimulus intervals. So first of all, you can see that your average ISI can go down very much lower, down to 5 seconds or 6 seconds. And it peaks at about what the block design is. This is at about a block design detectability. And for detectability, it turns out that your ability to detect the signal, in other words, make a map of the activation, gets better as your stimulus duration gets longer, even when you're doing jittered ISIs. But it's much better than a constant ISI, and it's very close to block design, very, very close to block design contrast. What's interesting, though, so here's what's the really important take-home message here is that this is the optimal ISI if you just want to make brain activation maps, if you want to just make the maps from your time series. But let's say you know where to look. Let's say you know where to look, and you just have one area that you want to look at. You want to characterize the hemodynamic response as well as possible. Then it's a totally different paradigm that optimizes your signal detectability. Then it's always better to go as fast as possible, to have as brief of a stimulus duration as possible, and as brief of an ISI as possible. There you get a lot more samples around this hemodynamic response, and so it's optimized at about a stimulus duration of 250 milliseconds there. So if you know where you're looking and you're just trying to understand the nature of the signal from a region of interest, it's a very different optimization. And there you want to go as fast as possible. Okay, and there's other advantages of event-related that clearly came out. So this is some work by Rasmus Burns showing that a big problem with doing block design, especially when you have tasks that involve like speaking or other sorts of things that might involve motion, is that you might have stimulus correlated motion, and that messes up the signal in fMRI. And the idea here is that if you have the stimulus correlated motion that looks like a boxcar, and if you have your hemodynamic response, there's a lot of overlap between this and this, and they're not orthogonal, and they overlap a lot, and so that's a problem. At least in this timing, they're not orthogonal. This is typically 20 seconds on, 20 seconds off. If you give a very brief stimulus, though, the hemodynamic response doesn't kick in for another 5 seconds. And you can separate your motion effect from the hemodynamic response really easily. So you can have speaking in the scanner if you give a very brief stimulus. So here's an example of speaking. And this is just looking at the motor area where you did a block trial. This is 20 seconds on, 20 seconds off. And here's the motion signal from the edge of the brain that occurs with speaking. And here's the hemodynamic response. They look pretty similar. And this is the supposedly real from the motor cortex. Turns out if you do event-related speaking, the motion response still occurs, but it's very shifted. This is the average motion response. It's very shifted from the average hemodynamic response, and you can separate this out very easily. If you use this as a reference function, you get none of this. So completely separated. So you can get rid of motion effects that way. Okay, and this pretty much summarizes, in a paper also by Rasmus back in 2004, the paradigms. And so the way you can look at this figure, and you can study this more later as you download the talk or whatever, is that this curve is a curve of, this axis is sensitivity to motion. So if you're close to 0, you're minimally sensitive to motion. If you're up here, you're really sensitive to motion. But in this axis is sensitivity to the real signal, better bowl detection. So, and you can see where all the different stimuli are laid out. So block design follows this curve right here, and very clearly it's not exactly optimal. Super sensitive to motion with a stimulus duration of 6 seconds, it's off at 6 seconds. But if you go up to about a stimulus duration of 10 seconds and off for 10 seconds, then that motion signal becomes exactly orthogonal to the hemodynamic response. And so therefore, you're both sensitive and completely insensitive to motion with a block design. So that's where it's good. With event-related, with varying ISI, with jittered ISIs, with the longer ISIs, this is stimulus durations of 5 or 9 seconds. It's not that good, but with a stimulus duration of about 4 seconds, with jittered ISI, you have a nearly optimal, nearly as good as a block design right here. And then event-related with constant ISI, sure, that's not very sensitive to motion, but it's not very sensitive to bold either. It's not very good overall. All right, and that's where you want to be right here, I think, is minimally sensitive to bold and as sensitive as possible to everything else. So here's just some maps on threshold showing an example of that. Here's block design. You have It looks awful. This is overt responses. This is talking in the scanner. Block design with 10 seconds on and 10 seconds off. Nearly no motion artifacts. This is event related with varying ISI with a 1 second stimulus duration, which is about here. You see that a little bit less sensitivity, but no motion sensitivity. As you can see, that's right along the curve of 0. And event-related varying ISI with 5 seconds. You can see that with 5 seconds, you're about here. I'm sorry, with 5 seconds, you're about right there. So that's also good. And you might be a little bit sensitive here and there to motion. And event-related with constant ISI, 1 second stimulus duration, 15 seconds ISI. This is where you're really insensitive, but yet you're not sensitive to motion. But you can see the curves are relatively flat. I mean, you can see these maps, qualitatively, they look, except for the one on the left, which is super just artifacts with motion, they look kind of similar. So, there's a weak optimization curve, essentially. Okay, fMRI adaptation effects. So first of all, the hemodynamic response is nonlinear, and this is what I've gone over before, is that essentially when you give a very brief stimulus, the black curve is what actual measured data show, and the red curve is what you'd expect from a linear system. And eventually when it reaches 20 seconds on and 20 seconds off, they match each other. But within this range of 0.25 seconds to 2 seconds, it's highly nonlinear. And the idea then is that you get a large transient response and then it habituates really quickly. So that's kind of the general concept that we're trying to exploit with other cognitive tasks as well. So here's also an example of how the neural input, the neural activity might habituate to create these nonlinear hemodynamic responses. You get a very large transient at the beginning, and then it quickly habituates. This is some really nice work by Nikos Logothetis, where he simultaneously measured EEG or single unit electrophysiologic measures and fMRI responses. And he gets this massive EEG response or electrophysiologic response, and then the hemodynamic response kicks in. And so it's very nonlinear in the first 2 seconds, probably because of this large neuronal transient, potentially. Okay, but the idea here was taken further by a number of people. And here's the basic paradigm, and where you have two different types of stimuli. If you give a stimuli, for instance, of one pool of neurons and then a different pool of neurons, you'll get the same bold response, even if these are close together in time. Okay? So, but let's say if you give a stimulus to one pool and then you give it to the same pool again, you'll get less of those neurons. They saturate a little bit and they become less active. And so you get less of a response. And so if you have a mixed pool, then you'll also, here's also showing the similar type of stimulus to the other pool, similar type of stimulus to the other pool, you get a decreased response as well. So relying on that, you could actually start to differentiate which neurons or how many neurons respond to certain types of stimuli. And people have done it not only for basic visual stimuli, but for more higher level processing. So this is cat faces. This is looking at cars versus other things, cars and random things as well, horses, cars, whatever. And so basically these are the different types. This is all the same. This is 2 different. This is 3 different, or 4 different. And this is up to 8 different stimuli. And what you find is that, first of all, with the same stimulus, with the cat faces, the signal quickly habituates. With the second part, with the intermixed stimuli, it habituates more slowly. So then you know there's 2 different pools of neurons here. There's a pool of neuron that responds selectively to cars, there's another one that's selectively different to the other ones. But if they're similar, if they activate the same neuron pool, let's say you have 2 categories that activate the same neuronal pool, then you might get this sort of response. They might still be activated. So this is a way of differentiating within a voxel what areas are active versus what areas are not. And so finally at the very end, where you have all the stimuli are different, then you get a very minimal habituation of the signal. Okay, So that's called fMRI adaptation. It was pretty much pioneered by Kellett, Grille-Spector. So if you look at any of her papers around 2001, you'll see this type of paradigm. Okay, orthogonal block design. Slightly esoteric because, like I said, you can orthogonalize different functions with a hit in contrast to noise, and people haven't really figured out how to design multiple orthogonal functions, but one person has. So early on, Susan Courtney had a really clever design in Nature paper. This was, oh, I didn't put the date. It's early 2000s or late 1990s, but basically it was a working memory paradigm in which she designed into the paradigm 6 different orthogonal input functions. So she was able to separate out within one paradigm, and that's actually it's desirable to do this sort of thing. It's always desirable to pack in as many comparisons as possible in a single time series. If you compare across time series, you lose a little bit because you have drift in time, drift in the subject, things like that. So your desire is to try to elegantly pack in as many comparisons as possible, like hence the continuous phase modulation. And here's another one. So okay, so she was able to, compare faces and houses, compare with maintenance phase, anticipatory delays, encoding versus recognition, all kinds of slightly different comparisons. They were all orthogonal to each other, and you're able to make these maps then that correspond to those specific functions that you don't have to worry that they're overlapping, because by definition they should be orthogonal. There might be some issues with the hemodynamic response causing delays that don't exactly cause it to be exactly orthogonal, but it's minimal. So you get very nice, clear separation of the various parts that are activated according to those tasks. So I'd recommend taking a look at that paper just for illustrative purposes. So free behavior or naturalistic or continuous stimuli is sort of where fMRI, I think, is starting to go, or it has been going for a while. I think it's still going in that direction. And everyone from Eleanor McGuire, who looked at like a naturalistic sort of navigation through a virtual reality. Yuri Asan has pioneered this, looking at if you're viewing a movie and listening to words, you can reverse order like the movie segments, and to see what parts of the brains are processing just the visual stimuli versus the semantic story that's being built. Jack Gallant has done this, where he has encoded movies and auditory inputs with multiple regressors corresponding to specific aspects of those movies or auditory, like, various objects or semantic representations, and then making reference functions corresponding to how those vary over time, and then putting in all those reference functions and pulling out the parts of the brain that are corresponding to those free behavior sort of movies. Early on in the late 90s or early 2000s, there was a DARPA competition where people were giving training sets of data where a subject was viewing a movie. It was a complicated movie, and they were showing the activation, and then they had to guess what the subject was viewing in like 3 other time series. And people were able to guess up to, I think, above 80% accuracy of what the subject was viewing in terms of the movie. So that idea is that the movie was the free behavior, and that was also using a combination of free behavior and decoding. And then Ryder Goebel and Christopher Ducharmes have been pushing, and many others have been pushing, the idea of using real-time feedback, which is a form of free behavior design, where you are told to increase a certain part of your brain activation or create some sort of brain activation pattern. And then you're given feedback by how much your brain resembles what the desired template is. And so people try to upregulate or downregulate, and they can do that. So here's the example of Eleanor McGuire, where she had people navigating through London, and this was broken down by various categories of what they were doing at certain specific times. And this was the virtual reality setting. And here's potentially what the subject was thinking. So you actually had them doing the driving and color-coded according to these very specific categories of what they were doing in the task. And then you simply bin the data, and you can pull out very elegantly the corresponding brain activation to those specific tasks right here. Another beautiful example is a little bit less related to humans, but I always like to bring this up because it's one example of a really nice multimodal integration. So this is work by Nikos Logothetis, where he actually had monkeys in the scanner. They had electrodes in their hippocampus, and they were sleeping. And every once in a while, during sleep, the electrodes would spontaneously record hippocampal ripples. So the ripples would be recorded. When they were recorded, he would bin the data and do fMRI analysis on the hemodynamic response. And what he found was pretty remarkable. He found that with corresponding to every ripple, you get a massive increase in cortical activation and a massive decrease in subcortical activation. So this sort of happens, and this, he's trying to build a hypothesis that this is how memory encoding happens. So when you're sleeping, your hippocampus sends out these ripples, it gets encoded in the cortex, and then the subcortical areas are kind of turned off for whatever reason when this happens. Okay, that's a naturalistic behavior as well, because you can't control when the ripples happen. You just have to measure them. So, and here's the idea of real-time analysis. So you have, the whole point is that you have the brain activation goes into some sort of collapsed representation that then's fed back to the subject, and that's neurofeedback. And then you have all kinds of other effects to optimal feedbacks and things like that. So this is Rainer Goebel's sort of platform in Brain Voyager, in which he does real time. And so here's the whole thing. You essentially have the subject in a scanner. They're trying to increase a bar or decrease a bar. You have the analysis being done in real time, and then that's feedback to the subject. Okay. So, and it's very important actually in real time, and this is an example of real time where people have tried to do real time initially in a way that was where they would continuously see the feedback and then continuously try to do something. But because the hemodynamic response is delayed by 7 seconds, it's a dual task, and so it's really hard to do that. So he actually had the insight to divide it into a block design. So you do the studies, you try to control your brain for 20 seconds, and then you wait and look at the feedback. You try to control it for 20 seconds, you wait and look at the feedback. That makes it much cleaner, and it gives a much more high fidelity response. So, and he was actually able to, this happens to be with patients with depression. He identified the network that's involved with emotion, and the subjects were asked to upregulate that network. They were asked to increase the activation in that network. Without going into detail about what the network is, the paper's right there in PLOS One. And within 3 runs, they're able to pretty consistently upregulate this network. And it actually had real behavior results. Not only were they able to upregulate the network, I mean, he's done other real time where people have played Pong in the scanner and things like that, but just using their thoughts, but that didn't really have behavioral translation. So here, it translated into a clear improvement across almost all the subjects in the depression score. And this is actually, it's one of the first clinical trial studies, and its first clinical trial studies in the UK. It has gone on, I think it's going on now. So it's actually being used clinically. Another example is pain regulation. This is some work by Christy Charms, in which he found the areas of the brain initially that corresponded to pain, selective pain or attention to pain. And there's amygdala, there's other areas as well. And he actually asked the subjects then, and he actually had a plot. Instead of a bar graph, he had a plot of the signal just from those areas. And he asked the subject to simply decrease the signal in those areas. And it was represented by, actually, this was not even shown to the subjects. This was shown to the subjects. The amplitude of this was represented by a flame. Either it was low or it was high. And that was it. So if the flame was low, they were doing a good job. They were down-regulating their pain perception. And it turns out that after they got out of the scanner, they actually did this. It took them several weeks to figure out how to do this. And they actually devised their own strategies. They get out of the scanner, and it turns out that their perception of pain, the change in their perception of change corresponded pretty nicely to how well they were able to downregulate the signal. So and it seemed to last up to 6 months afterwards. Okay, so the last 15 minutes, just going to quickly go over resting state and fMRI decoding, if I can do that. All right, so even So before Bharat Biswal had his discovery in 1995 of resting state fMRI, people did know that there was a lot of physiologic noise in the signal. And as I mentioned before, this is sort of the predominant physiologic noise. If you actually have signal to noise over time, it reaches a peak at about 100 to 1 or so. And so that's a problem. And there's a lot of physiologic noise in this signal. Otherwise, it would go straight at a 45-degree angle. And there's a lot of contributions as well. Everything from the stuff we don't want, like brain pulsation and CSF, vasomotion, changes in B naught, the magnetic field with lung expansion, bulk motion, scatter instabilities, changes in blood CO2 with changes in breathing depth. What we'd like to see is only the changes in spontaneous neural activity. So that's what we're always looking for. That's the battle right now, of actually trying to get rid of all this stuff. We're not there yet. All right, so just a quick showing of what I mean by resting state. So as I showed you before, this is the signal from the left and right motor cortex in which the subject is tapping their left fingers, then right fingers, then left fingers, and right fingers. And you can see the corresponding out of phase signal from left and right fingers, 180 degrees out of phase. Okay, and here the subject is lying in the scanner, actually doing absolutely nothing, not tapping their fingers, not doing anything. And the signal looks like noise. But it turns out that if you took a seed voxel in the motor cortex and used that as a reference function, and then calculated the correlation coefficient of that voxel with every other voxel in the brain, you'll get the other motor cortex. So even when you're not moving your fingers, the functionally linked, connected cortical areas are talking to each other, supposedly. And there's a lot that goes into this interpretation, but essentially the raw signals are correlated. Correlation coefficients are at about 0.3, as opposed to here they're about 0.8 or something like that. But and here's Bharat's experiment, same thing I just showed. This is tapping their fingers versus just resting. You get almost the same areas, not quite exactly the same. Okay, so resting area fMRI, resting state fMRI. Why is this important? Well, the number of papers has exploded. Here's an example. Actually, this doesn't quite do it credit, but what's really interesting is that when Bharat made this discovery back in '94, everyone kind of ignored it. They were really busy with their developing fMRI task activation. And only at about 2006 did people look at this again. 10 years later, they kind of ignored it. And then they said, oh, maybe this resting state stuff is interesting. And then the papers started taking off. And so now it's going up exponentially. Right now, it's probably about, I would say, over 1,000, easily over maybe almost 1,500 papers a year, just in resting state, almost half the field. If you go to OHBM, it's at least half the meeting is resting state. And here's just looking at the fraction of connectivity papers. Right now, it's at about probably 30%, I would say. Okay, and what we're finding is that there's a lot of disorders, either neuronal or psychiatric or developmental disorders, that seem to manifest as showing altered connectivity. One of the big problems in using fMRI to understand disorders has been to find the right task. I think for the last 10 years, we had a group here studying schizophrenia in which they're doing an end-back memory task, which is a great task, and it differentiates people with schizophrenia and not, but it's just one dimension of probing in a huge, huge space. And so how do you know that task is really going to get you the essence of the difference between schizophrenics and normals? You don't. Resting state might capture, with a little bit wider net, these differences. So, and also it's nice because you don't have to have the subject do anything in the scanner. So, it's really hard to get patients actually to do tasks. And that's one of the big confounds, that they do it either worse or they don't pay attention, they move too much, whatever. You just have them lying in the scanner doing nothing. It would be great if that completely worked. So the methods themselves are in their infancy. We don't, in spite of, there's people developing packages, FSL, SPM, AFNI as well. And of course, all the creators of the packages will be very confident that this is the way to process resting state data. It's totally untrue. There's many different ways to process resting state data. There's no correct way still, and it's still evolving, because we frankly don't really understand the signal. So So that's still evolving rapidly. And the neural correlates of spontaneous fluctuations aren't fully understood. This is also why the field is so important. There's people who are trying to understand really what are the correlations between this 0.1 hertz fluctuation signal and the rapid communication that occurs between neurons. What is this relationship? We don't know that. Okay, but there's many early processing methods for looking at this, either using sort of an hypothesis-driven method of picking a seed and seeing what correlates with it. This is some nice work by Mike Gracious, in which he was the first to discover not only that the default mode network previously discovered by Mark Rakel goes down during all cognitive tasks, but it also shows during rest a modulation in the signal. And this is some nice work by the Oxford group, early work, using ICA to break down the signal into corresponding patterns. It's important to note, though, I always like to emphasize here that when you do ICA on your data, these networks that are broken down are not, they might be, but they don't have to be the actual neuronal networks. What ICA does is impose a spatial independence of the signal. So these are networks that have a spatial independence imposed on them. So there might be a lot of overlapping networks that are spatially dependent that you don't see using ICA. Okay, so it's important to keep that in mind when using ICA, that what you produce with that isn't necessarily neural networks. Okay, one other fun thing to do with seed voxel approaches, though, you can make movies of, as you pass the seed around. There's a function in AFNI called 3D T-core, which you can have a seed and look at the correlation with that seed in real time. This is something where we actually looked at the motor cortex and passed the seed right along the motor strip. And as you pass the seed along the motor strip right here, you can see the corresponding opposite motor strip showing activation. This is with a sort of gyrified map, and then this is a flattened map right here as you pass the seed along right here. And then you can get information from this. So looking at this as we pass the seed along the motor cortex, we can see there's some oscillations in what's correlated. And here's the same sort of thing, which is actually a nice demonstration of the fidelity of connectivity. So what it shows here is that as you move the seed voxel, you find that the corresponding, so this is a breakdown of the motor cortex from the foot, the toes to the tongue right here. And you find that as you move from the toes to the hand to the tongue, you find a modulation of connectivity. And from more track tracing studies, more anatomic tracing studies, it's pretty well known that the hand area has the least connectivity across hemisphere relative to the other areas. So that seems to sort of confirm that what we're looking at seems to correspond to actual connectivity and not just some artifact. Okay, how do you also look at it? Other ways of looking at it. One way is to break the brain into a bunch of segments and then look at the time series of each of these segments and calculate the pairwise correlation between each of these segments and put it into a matrix. And you have that here. This shows in one big collapsed form what parts of the brain are talking to the other parts of the brain. And you can divide this into different chunks of time as well to show, okay, maybe if you just do it across 2 minutes, the areas are talking in a certain way. Across all 30 minutes, they're talking in a different way. Here's basically also another way of looking at things. This is some one of the movies from the WashU group that shows if you can put balls and sticks and look at connectivity of this pairwise correlation matrix and put it in spatially deconstruct it so that each one of those segments is now a little ball, and you can see the correlation as they connect. And for me, it kind of gives you a gestalt of what's going on, but it doesn't. It's hard to interpret these plots in any sort of detail. I think it's actually much better just to keep it in that pairwise correlation matrix, so you can actually then compare the patterns of the pairwise correlation matrix. So this is nice, but it's good for maybe a movie, but not for actually, I feel, for real analysis like this. So, and like I said, you can actually divide the brain. You can divide the brain into specific segments and look at networks and how the networks correlate with each other. And so this is also looking at the dynamics of how the correlations change over time. And this is, I like this sort of plot where it shows over time, if you have a sliding window in which you look at the pairwise correlation matrix, how that evolves, you can see that the correlation between these various networks is constantly fluctuating. And so there's two things you can get from that. One is where they're maximally fluctuating, and two, the time constants of these fluctuations. There's been some work on how to clean up the time series data. What we've done is what I've considered actually a sort of a goal of resting state. So not only get the really strong connected areas, but to try to, within one run, make an entire map of brain connectivity. And so we're almost getting there. This is an average of 7 subjects. And this is where we break it down, one time series, one 10-minute time series, into 350 clusters. And you can actually see now, if you look at all the areas that are most correlated with themselves or with other areas, you can see Broca's areas, you see Wernicke's area, you see the motor area, you see sensory, other language areas, visual areas, all seem to organize themselves according to what you'd expect. And you can start to break this into a functional map of the brain, looking at purely resting state. It's across all the brain. Okay, and it does correspond. I won't go into detail here, but basically we did a very basic experiment of toe, hand, tongue, language, and faces. And the functional units do correspond to the areas that show up during rest as well. So they seem to show a fidelity or high fidelity correspondence to, like for instance, here's one person that is left dominant, and also in the task he's left dominant as well. And you get face areas as well. This is all the face areas seem to show up really nicely too. Okay, global correlation. I think I'll skip this. You can actually look at this as basically an issue of how do you clean up the noise. And so one issue in resting state is that, do you get rid of global signal or not? It's a huge issue. And for the sake of time, I'm going to skip over it right now. And I'm going to go right into fMRI decoding. Just a brief introduction. The simple idea here is historically, it started with Jim Haxby, I think. It could be traced back to him when he was here at the NIH. He's an expert on face processing. And he had a beautiful experiment, which he actually did an experiment comparing faces versus houses. And he found a pretty clear differentiation of large face blobs, and the houses were a large blob. And so he's like, okay, let's take this one step further. Let's look at other things, chairs versus shoes, for instance, things that may not have a big blob of activation corresponding to them. And what he found, okay, so this is showing the data that from the naked eye, if you're looking for blobs corresponding to the chair blob versus the shoe blob, it's hard to actually differentiate this. It becomes more of a mess. There's more high-frequency information there that your eye doesn't pull out as easily. And it starts to look like noise. But what he did was something, instead of giving up on this data, instead of looking at it and saying, wow, this is really noisy, I'm just going to throw it away and find something that's simpler, he actually did something really clever. He split the data into 2 halves and calculated the activation map for one half and then the activation map for the other half, calculated the spatial correlation between the activation maps, and found that for the same type of stimulus, the spatial correlation was much higher than if you compared a different type of stimulus. Even though, shoes are, it's all visual cortex, but then there's different patterns riding on top of this activation that are much more subtle, that the human eye can't really discern that well. And if you try to make activation maps comparing them, it looks like salt and pepper. But if you actually do the actual spatial correlation, it pulls it out. So what did you do? You didn't actually map the areas that were showing the correspondence to a shoe or a chair. All you did was decode. You were able to tell what the subject was looking at the time without making the actual map, because the map is too messy, essentially. So Nico Kraigastorti picked up on this, so he put in 96 different stimuli and calculated the spatial correspondence, the spatial correlation between these stimuli and put it into an entire matrix called a dissimilarity matrix. So these are the visual stimuli right here, everything from guns to signs to faces to animals, whatever. And he divided them up into human, or animate versus inanimate, human versus not human, body, face, and actually found that looking at inferior temporal cortex, IT cortex, in the 1,000 most responsive voxels, the patterns showed a clear differentiation that organized themselves between animate, inanimate, human, not human, and so on. So he was able to pull out this effect without having to sort of rely on picking the category and sort of averaging them all together and mapping them. It was a kind of a looser, more loose sort of paradigm design that allowed for different individual objects to show their individual responses. And what was nice is that he did try to do the same grouping for early visual cortex and found, oh, the early visual cortex doesn't show this pattern whatsoever. So human IT categorizes based on semantic content, it seems, and early visual cortex doesn't. And this is clearly shown right here in this sort of pattern effect. This is a sort of decoding. He compared humans and monkeys. And in the monkeys, he didn't look at fMRI response. He looked at 674 different single cell recordings and made a pattern activation map from those. And what he found was a really nice comparison without having to do any topography, just simply knowing that you're looking in IT cortex, that the patterns corresponded almost exactly between different species and also different modalities, using single unit versus human. Okay. And then there is a question of whether or not the patterns correspond to something subvoxel. Like, for instance, the one hypothesis is that you have, let's say you're trying to decode visual orientation. You have this pinwheel activation. There's some voxels where you might have a slightly more number of certain orientation columns than others. In other voxels, you might have slightly more other types of columns. And so if you do this, those will be the encoding voxels for that pattern. So that's what basically Kamatani and Tong did, showing right here. So basically did different orientations, really big voxels at 3 Tesla. And while the map itself is almost meaningless, this happens to be an arbitrary representation of the voxels as they chop up the brain, and those voxels, whatever happens to be the predominant orientation column, is the encoding column. And then using this as a template, they're able to tell what orientation the subject was looking at. So once again, the map is nearly meaningless. It's what you can tell about what the subject's doing that's important. So, but then the other hypothesis is that there's some low frequency sort of macroscopic effect that's really driving all this. And that's still probably up in the air, and there's a debate going on between the both. It's probably both in different cases. Okay, so with that, I'd just like to end. I mean, there's a lot of other, the field of having different input strategies is evolving pretty rapidly. This at least lays down some of the basics. And I got a little bit less into processing strategies, but there'll be a lot more talk about processing during the summer. Okay, any questions? Yes?
So near the beginning of your talk, when you were talking about potentially testing different jitters on the start of the hemodynamic response function, and also testing different hemodynamic shapes of the function. So what's the trade-off of doing that versus then overfitting your data and potentially finding false positives by just testing a bunch of different HRFs? Yeah,
that's a good question. So the question is, right, is what's the advantage of, I have to repeat this because this is being recorded and there's no microphone. So the question is, so the advantage so you're saying that if you put in different response functions, and that worry of overfitting versus,
right.
Yeah. So I think we're referring to the one where I showed maybe 10 different response functions. And
right. Well, I understand, where if you collect 9 hours of data, right, then you can be pretty certain that it's not a false positive. But...
Right. Yeah, exactly. So I was going to say, right, if you have high enough signal to noise, then you don't have to worry as much about overfitting. But I think you're right, and I think this as an example, right, there's always the risk of having some aspect of the noise or motion separate what looks like a hemodynamic response from something, or maybe divide up the hemodynamic response artificially and say, oh, well, there's clearly something interesting going on here because there's an early latency. So one example, I think it's not really a worry as much in this case of overfitting, but rather of overinterpreting something. So for instance, what people did in the early days was they were trying to infer too much from the latencies. So they were saying, oh, well, these parts of the brain show a 2-second, they happen 2 seconds earlier than this part because this hemodynamic response shows the phase shift. And in fact, it was purely plumbing. It was like just because that part of the brain happened to have more capillaries than another part of the brain had more vessels. And so that was it wasn't a problem of overfitting. I think that if you do try to fit and match the latency, that boosts up your power. But if you try to say, oh, something happened early, therefore it's an earlier part in neural processing, that's overinterpretation of that. But there is the other issues of overfitting that I don't really feel that this aspect of what I was talking about got into too much. But you're right, there is the issue of overfitting though too. But yeah, I don't know if that answers completely, but still. Okay. Okay. All right. Thanks. And the next talk then will be Wednesday, and that will be back in Building 40. So at 2:00. Thank you.